Hello, good afternoon, good evening, good morning, everybody. Today we can explain some of information about bacterial classification. So, we will start with how are microorganisms classified. So, to answer these questions, we do have simply taxonomic ranks that we can classify microorganisms and all organisms in the world according to such taxonomic rank. And then we can go for bacterial nomenclature, how we can name the bacteria. So we do have a group of criteria such as the biochemical properties or biochemical test for the bacteria and the physiological properties, genetic properties, and also the morphological shapes of the bacteria. So let's start by the taxonomic ranks. Microorganisms and all organisms can be classified according to such criteria, starting by the kingdom. The kingdom is, for example, for the bacteria is a prokaryotes, and then we do have the division, and then we give example for the division as the Gracilicutes, and then class. The class, for example, Scotobacteria, and then the order Eubacterialis, and family, example Enterobacteriaceae, and then the genus Escherichia, and species E. coli, and subspecies also we can include Escherichia coli 0157H7. So such taxonomic ranks are general for all organisms to classify. Let's start by the bacteria. We have mentioned that the bacteria usually classified according to numerous of criteria, including first, the physiology of bacteria. The physiology of bacteria means how the bacteria grow in culture, including the pH preferences, the temperature preferences, the type of culture media. We do have three basic types of culture media for the bacteria. For example, we do have non-selective media that is general for most types of bacteria, and we do have the selective media that uh, allow the grow or only specific type of bacteria, for example, gram-positive bacteria or gram-negative bacteria. And we do have the differential media that differentiated between two classes of bacteria. For example, differentiates between gram-positive and gram-negative bacteria. So here the picture explain some examples about the differential media. The first one is this one, the AMP, that is a specific media to grow E. coli on it. And the second one is the McConkey, that can differentiate between lactose fermenter and non-lactose fermenter. So this is the differential media. And the second criteria that we use to classify bacteria is the morphology of the bacteria. So we can usually see the shape of the bacteria by using staining method, for example, gram staining, and by microscopic methods, advanced microscope or the general light microscope, we can identify the shape of the bacteria. So we do have two basic groups of bacteria according to the shapes. It is gram-positive and gram-negative according to the stain, and we do have the cocci according to the shape and rods also. So the other criteria is the biochemical test. We do have enormous biochemical tests that we can use to identify the bacterial at genus level or at the species level. For example, we do have the oxidase test that used to detection the presence of cytochrome C, and usually it is used to identify oxidase positive bacteria from oxidase negative bacteria. And also we do have the catalyse test that catalyze the reaction of hydrogen peroxide into H2O and O2, and usually is used to identify staphylococci from streptococci. We do have also the coagulase test, the enzyme that produced by specific type of bacteria to coagulate the plasma of blood. We do have the citrate also test to identify citrate positive and citrate negative bacteria. So we do have other biochemical tests that used to identify the bacteria, and we can run such biochemical tests either by using standardized kits or by using standardized machines. So also, this is an example for using the biochemical test to identify or classify the bacteria. So for example, we do have a staining method and identify the bacteria as a gram-positive cocci. And then we are running the catalase test. The catalase test identify two groups of bacteria. For example, catalase positive, staphylococci, and catalase negative is the streptococci. And then we do have the coagulase test for catalase positive group. To identify the coagulase positive bacteria, which is Staphylococcus aureus, and the negative bacteria, that is Staphylococcus epidermidis. So this is the example for using the biochemical test to identify the name or identify the species of the bacteria. And we have lastly the genetic characteristics for the bacteria. So to identify the genomic materials for the bacteria, is it chromosomic materials or it is a plasmid materials? We do have numerous or many gene identifying methods. For example, we do have the PCR, polymerase chain reaction method. We do have the next generation method to identify the genomic content of the bacteria. And by using such methods, we are able to identify, for example, antibiotic resistant genes of the bacteria. Also, we can identify the genes encoding enzymes. So such genomic characteristics are more advanced to identify the bacteria. And also we can identify the specific genes of the bacteria, for example, the 16 sRNA, because it is conservative among most of the bacteria. And lastly, this is only basic information about bacterial classification. And if you like the content, you can visit the website www.wordpress.science135766799.wordpress.com. And thank you for your watching this video. If you like the content, you can subscribe to the channel for more upcoming videos.